This video is created for teaching purposes only and is a visual representation of a general hematology complete blood count analyzer, also known as a CBC analyzer. It is not meant to represent any specific manufacturer or model. As such, the order in which analysis takes place and the type of method of analysis is not necessarily reflective of any specific analyzer used in the clinical setting. Room temperature, EDTA anticoagulated, whole blood samples are loaded onto the analyzer using the rack provided by the manufacturer. This sample barcode, applied during specimen collection, contains the patient's identification, demographics, and test panel as requested by the physician. Examples of test panels include CBC only, CBC with differential, and CBC with reticulocyte count. Each sample is then mixed thoroughly to ensure even distribution of cells within the plasma. An aliquot of whole blood, we'll call AWB, is obtained by needle insertion through the cap. The volume of this aliquot will vary from model to model. The aspirated whole blood sample, AWB, is split into 3 different aliquots for the 3 major parts of the CBC analysis. 1. Red blood cell and platelet counting and measurement. 2. Hemoglobin measurement. And 3. White blood cell counting and differential. We will refer to these as AL1, AL2, and AL3. The first aliquot, AL1, is delivered to the platelet red blood cell counting chamber. AL1 is diluted to make it possible for the cells to flow through the aperture single file. A direct current electrical circuit is applied to the aperture, and when the cell passes through it, impedes the current. The larger the impedance, the larger the cell. This methodology was pioneered by Dr. Coulter in the 1950s and is known as the Coulter principle. It is still in use today in almost all current CBC analyzers. This method is used to both count and measure the size of the red blood cells and platelets. The 2 cell populations are separated by size and counted accordingly by preset thresholds programmed by the manufacturer. The resulting data is recorded on histograms and are displayed on the analyzer printout. A second portion of AWB, AL2, is diluted and then combined with hemoglobin reagent in the hemoglobin reaction chamber. This reagent contains several chemical compounds which first lyse the RBCs to release the hemoglobin and then reacts with the hemoglobin in several steps to change the color from a rusty brown to bright cherry red. This color is what is measured spectrophotometrically and is directly proportional to the amount of hemoglobin in the sample. The third aliquot of the AWB sample, AL3, is first diluted with diluent and then combined with specific volumes of several different reagents. The first reagent added is a red blood cell lysing reagent to eliminate red blood cells from the fluid. The next reagents added make the white blood cell membranes more permeable and then stain the WBCs with various dyes to make them easier to identify. This prepared dilution is then injected into the flow cell and using flow sheath technology, the white blood cells pass through the laser light single file. Depending on the complexity and size of the white blood cell, the light is either scattered in all directions or will partially pass through the white blood cell. All the reflected and refracted light is detected using various photodetectors and analyzed by algorithms built into the analyzer software to separate the white blood cells into their different cell types. This information is represented by the scatter plot that is displayed on the analyzer printout. This same process of white blood cell identification is also used to identify reticulocytes. Different reagents are used, which will stain the residual RNA in the red blood cells and can then be detected using the same laser light in the flow cell. This analysis is only completed if a doctor has ordered it in the test profile. Each analyzer manufacturer will have unique reagents and analysis techniques and algorithms to identify white blood cells and reticulocytes. The principles of electrical impedance and radiofrequency to determine the white blood cell count and size may also be applied at this stage and is not shown in the video. Each dilution chamber, reaction chamber, and analyzing component is flushed with diluent. Once analysis is complete and prior to the next sample to be aspirated, this prevents carryover contamination from one sample to the next.